All right, so what is metabolism? I want you to think about your answer for that. You can pause it. So most students will say in class metabolism, what we relate that to is they're going to say, well, it's how much weight I gain or how much weight I can lose. And that is partially correct, but it's only about halfway correct. Metabolism is the sum of all chemical reactions in the body. So it's not just how we break down and assimilate nutrients, all right? But it's also, sorry, I'll turn that down. Let me heard that big old noise there. It is also how we make things in our body, okay? And so this is broken down into two portions, catabolism and anabolism. And so that's what we're going to be looking at. So catabolic reactions are catabolism. Metabolism is going to use enzymes. Okay? Enzymes are really, really important. So those are going to drive the reactions that are going to break down or catabolize organic molecules. Remember, catabolism, think catastrophe. We are tearing things apart, breaking things down, and releasing energy. And so we're able to utilize that energy from those catabolic reactions to do anabolism or to make things for anabolic reactions. So we can make larger, more complex molecules that we need to do other processes in the body. Okay. So the reducing power comes from the electrons that are available in our little carrier or shuttle molecules, and that's going to be NAD and FAD, nicotine, adenamide, and then flavin. And really, you don't need to know the words so much as you just need to know NAD and FADH, and then those are going to be changed to NADH and FADH2 as we go through oxidation reduction. Okay, so NAD and FAD are in their reduced forms here. All right, and energy is needed in a large quantity for these anabolic portions of metabolism. We got to have energy to build things to assemble. Remember, anabolism is assembly. Okay. Um, and then that energy is stored in those bonds, and so they're able to carry that energy elsewhere. So here's the portion of metabolism we're thinking about when we hear the word metabolism. Metabolism requires nutrients from the environment to be broken down into what we call precursor molecules. So we can send them into these three different pathways for organisms that they can use. Okay, so these processes are varied, especially in bacteria, but generally they're going to fit into these three basic catabolic pathways here. Okay, often glucose is the nutrient. Just don't fall into the trap of thinking only glucose goes through these processes. It's just the main way, so that's the way we teach it, and that's what we focus on. Okay, we'll talk about some of the alternatives as well. So the pathway is usually going to be related to the organism's oxygen requirements. Okay, so we have aerobic respiration. That would be an organism that can utilize oxygen. Then we're going to have anaerobic respiration, and that's going to be an organism that can't utilize oxygen. And then, of course, we're going to have fermentation. All right. And so here we've got aerobic respiration, and that has the processes of glycolysis, which is going to it's going to make 4 ATP, but then it's going to give us 2 net because we have to kind of prime the pump and put 2 ATP in, and it's going to give us 2 out and then we're going to move and take that product from glycolysis, all right, and move it to the next step here, which is going to be Krebs cycle, okay? And it's going to break this down a little bit further, and it's going to send all these electrons through these little carrier molecules here, our NAD and FAD here. These are going to carry electrons and hydrogens to this electron transport system, which if you've had anatomy, you have heard plenty about all of this, all right? And then we're going to utilize oxygen as the final electron acceptor. Something has to hold on to those electrons when we're done, okay? And then we're going to make a total of 36 to 38 ATP with aerobic respiration, okay? Anaerobic respiration, and this is going to be like what you would see in bacteria. It's very similar, okay? The thing is, instead of the final electron acceptor here being oxygen, it's going to be a little more varied. We've got sulfate, nitrate, and carbonate here that can take that oxygen, okay? And it's going to make anywhere from 2 to 36. It's very varied. And so this is why we say that bacteria and other prokaryotes are structurally simple. They're one cell, their structures are very simple, but they're metabolically complex, meaning we have to have oxygen as our final electron acceptor for aerobic respiration. This is why we breathe, is because we've got to have oxygen for aerobic respiration. Okay, but bacteria are more capable of using other substances to take those final electron acceptors. All right, so we can use sulfates, nitrates, and carbonates, and so they're a little more complex that way. And then we have fermentation, and that's where we are going to pretty much just have a very small amount of ATP made. Organic compounds are going to be the final electron acceptor here, and this is where we make our alcohols and that type of thing. Like you can do alcoholic fermentation. All right. So anyway, we're going to get into more details for this. Okay, so here we're looking at aerobic respiration, and this is what we as a eukaryote predominantly do. We can do lactic acid fermentation, and we also have acid types of fermentation in our muscles if we run out of oxygen, but it's not very efficient, and so we rely on aerobic respiration. So we're going to have those steps of glycolysis, the Krebs cycle, and the electron transport chain, which is also sometimes called the respiratory chain, although that's kind of unique to this textbook. I usually just see it listed as electron transport chain, but they mean the same things, okay? So aerobic respiration here is a series of reactions. Glycolysis in and of itself has 10 steps. Krebs cycle has 8 steps. So this is separate metabolic pathways that we're linking together, okay? And so it's a series of reactions that convert glucose to carbon dioxide, to pyruvate, and it's going to allow the cell to recover a large amount of energy. Okay, so we use glycolysis, the Krebs cycle, and the electron transport chain. We're going to rely on that free oxygen. We've got to have that oxygen at the end of the chain to pick up those electrons and hydrogens, okay? Because if we don't, you'll see that we're going to lose our concentration gradient and things will just shut down. And so we're going to have a large amount. Remember that maximum is 36 to 38 ATP, and that's an average. It can range in there. And you're going to see things like bacteria, fungi, protozoa, and of course more complex animals are going to utilize aerobic respiration. Okay, so more on anaerobic respiration now. Anaerobic respiration is going to be used strictly by organisms that are anaerobic, so they don't use oxygen, or those who can metabolize, those who can do both, those who can metabolize with or without oxygen. And so we're talking about mostly about microbes here that can kind of do both. And so this is going to involve the same 3 pathways as aerobic respiration. So you're going to have glycolysis, you're going to have the Krebs cycle, and the electron transport chain, okay? But you're not going to use oxygen as the final electron acceptor. Something else is going to pick up those oxygens and those hydrogens, okay? And so that's going to be our sulfate, nitrates, and carbonates, all right? So they're going to be able to utilize those as part of their metabolism, okay? Now, fermentation is an adaptation by facultative and aerotolerant anaerobes to ferment, which means incompletely oxidize glucose, all right? And so oxygen isn't required, and then the organic compounds are going to act as those final, or you could say terminal electron acceptors, okay? So final terminal electron acceptor is the same thing, just different terminology, all right? So we're going to take a look here at glycolysis. In class, I draw this out, and we actually do this as an essay. And so I'm going to go ahead and find a link for that where I draw it out, and then I'm going to post that, and I'm going to post the like little templates that I usually give my students to use. If I have them, I'm pretty sure I have it for this. I'll probably do that for this and the Krebs cycle. Just because it's really, really important for our end students to know that process. It may take all the steps, like for instance in glycolysis, there's 10 steps. You would probably need to know all of those. Krebs cycle, there's 8 steps. Those could also show up. You need to really know this in a little more depth than this textbook goes into. So I want to give you that opportunity to watch that video because I step out the whole thing, and there's no need to really recreate the wheel, although who knows, I might decide to do it. But Okay, so, all right, we are going to look here at this. We are going to have the steps of glycolysis. We don't have all of them listed here, but I'm going to give you those. You're going to start here. Well, all three metabolic pathways, let me say, begin with glycolysis, which are going to turn glucose into 2 copies of pyruvic acid. So that occurs actually without the need of oxygen. Okay, and it occurs in the cytoplasm. We're not in the mitochondria yet when we're doing glycolysis. Okay, so that's going to occur in the cytoplasm. There are some differences in places in bacteria and things where these type of things occurs, and we'll look at that. All right, so it's going to take that glucose and convert it or break it down into 2 pyruvic acid molecules. So we're going to go from this 6-carbon glucose molecule. The first step is to convert it into the glucose, well, this is kind of skipping some of those steps, but it's showing you now you've got the fructose 1,6 diphosphate here, and then you're going to have the splitting phase. Okay, glycolysis is split into energy investment, energy payoff, and splitting. Okay, or splitting then energy payoff. All right, and so this is the splitting portion where you're going to break this down now into 2 molecules. So there's another reaction that is going to break fructose down, fructose 1,6 bisphosphate or diphosphate is what this one is listed as, into 2 3-carbon molecules. Okay, and then you're going to invest your 2 ATP here. By the time we get to this point, we have made 4 ATP, and we have loaded electrons and hydrogens onto the 2 NADHs, all right? And so we now have 2 pyruvate molecules. So there's 5 reactions there. So there's a total of 10 reactions, okay? So we have 3 here and 1 here. That's your first 4. Probably this is skipping the intermediate step, which is included. And then we're going to convert the 3-carbon molecules into 2 3-carbon pyruvates. And you're going to have a total of 2 ATP and 2 NADHs. All right, so we yield a net 2 ATP. Okay, and you need to know the inputs, which will be glucose, the outputs, which are going to be, or products, reactants would be glucose, products would be pyruvate, 2 pyruvate, okay? Some texts may say pyruvic acid, 4 ATP, 2 net, and 2 NADH. So you always need to do those yields. And now these 2 pyruvate are going to be the reactants in the next section of the pathway. So now some textbooks will break this down and they'll have like an intermediate step. Some textbooks include that in glycolysis or the Krebs cycle. And so when I draw this out, you're going to see that little short, it's called link reaction or intermediate step, okay? So don't be thrown off by that. It's just included in one or the other steps. Sometimes some textbooks just kind of lump them all together. Okay, so now we're looking at the Krebs cycle. This is also called, because it's science and not hard enough, for some reason they think we have to have 2 or 3 names for everything. And so the Krebs cycle is also called the citric acid cycle. In this textbook, you may also see, and also if you take upper level classes, the tricarboxylic acid cycle or TCA cycle. Okay, and so those all mean the same thing. So we're going to transfer the energy stored in acetyl-CoA. We're going to take that energy in the form of those electrons and hydrogens and put them again on some little shuttle molecules. We're going to use NAD and FAD, and then we're going to reduce them. That's the transferring of electron. Remember, oxidation is loss and reduction is gain. And so along the way, the acetyl-CoA is going to join with oxaloacetic acid and then participate in 7 other steps. Remember, I said there's 8 steps in the Krebs cycle, and you're going to get a total of 6 NADH, 2 FADH, and 2 ATPEs for each molecule of glucose. Now that means you're putting in those 2 pyruvate or pyruvic acid, okay? And this link reaction is where that's converted to acetyl-CoA, all right? So they're including that here in the Krebs cycle. And so it's going to be converted to that acetyl-CoA. And so for every glucose, how many pyruvate did we make? 2, correct? So this is going to turn 2 times. So you always need to be careful on an exam to pay attention to how it's worded. Now, I haven't pay too terribly much attention to. It's been a while since I made the test questions, but this textbook does not break it down and emphasize the per turn. So on this exam, well, yeah, actually it does. You have an acetyl-CoA with 1 ATP, 3 ADH, and 1 FADH, and 2 CO2s. Okay, and so the total then is going to be your 6. This is for 2 turns. So per glucose is this section here. This is per glucose. Ooh, this is so hard to write here. I'm going to do, right, glucose right here. This is per glucose. And sorry, this laptop is not, I can't use a pen on it, so I can't write real neatly. And then this is per turn. Okay, so be sure to keep that straight in your head. Per turn is going to be the smaller numbers. If it's per glucose, if you have the larger numbers than you need to know, you can just divide those by 2. But that's how you can kind of tell the difference, okay? And so that's per 2 turns for every 1 glucose, all right? And so your reactants are going to be here. It's going to be the acetyl-CoA, okay? And then your products are going to be your NADH, your FADH2, and your ATP. All right. And so all these other things in this step you're going to see are like oxaloacetate, citrate, isocitrate. There's going to be some really fun words in that when you watch that video. Those are all intermediate products in this particular portion. All right. I forgot to mention in the previous slide that sometimes things like that oxaloacetate may actually go to other pathways. And so sometimes these products that are made at certain sections, they can actually be taken off and used for other things if needed. Okay, it's kind of like if, well, if I have a saddle and a part breaks, I can go rob off of one saddle temporarily and borrow that. Okay. All right. So this is an electron transport chain. This is a chain of, basically these are proteins. They're special proteins that are embedded in the membrane, in the cristae membrane for eukaryotes, and the cytoplasmic membrane for bacteria. All right. And so these are going to receive electrons. So NAD is going to, oh, I can stick my pen here, come in here and dump its electrons off in this first one. Don't be put off by these crazy names like ubiquinone and NADH reductase, cytochrome reductase. You don't need to know these at this level. Biochemistry, maybe, okay, but at our level, you don't need to know the names of these. Now, this one on the end, this ATPase or ATP synthase, all right, those 2 different names for it, same thing. You do need to know the name of that one, all right? And so NAD is going to come and take those electrons and the protons, which is a hydrogen, okay, a hydrogen ion, that's why it has the plus, and dump them off here, all right? And then the electrons are going to be sent on down this pathway, and then the protons are going to be pushed across the membrane, which is really important, okay? FADH is going to come, and it doesn't show it, but it's going to dump its electrons off here, okay, or here, all right? So it's going to dump them off a little further down the chain. And so that's one of the reasons why we have that average of the 36 to 38 is because we get more out of NADH than we do FADH2. So it depends on the electron shuttle that is used. All right. And so we're going to receive electrons from those reduced carriers, NADH and FADH2, that we made in glycolysis and Krebs cycle, and pass them on to the chain of these molecules here, okay? And so this is going to allow the transport of hydrogen ions to go outside the membrane to this extracellular space here, all right? And it's going to become more positively charged. And this is where understanding those ion gradients is so important. This is going to create a higher amount of hydrogen ions or protons. They're kind of the same thing because a hydrogen is just a proton and an electron. And if it's a hydrogen ion, the electron has been stripped away. So it's pretty much just a proton, okay? The electrons go through here. They go through, sorry, you can't see that. Electrons are going to travel through here, okay, and be picked up down here by the final electron acceptor, all right? The hydrogens, though, are pumped out to this side, and so they're creating a gradient. They're creating a difference in concentration of hydrogen ions. It's going to be higher here than it is here, okay? That is going to create what we call a proton motive force, all right? Proton motive force. And that force is what is going to drive this particular enzyme here, this ATP synthase, all right, and it's going to cause it to turn. They're going to flow through here from their high concentration to their low concentration, okay? They're going to flow down their concentration gradient through this ATP synthase, I always say ATPase, which comes from another class. It's the same thing. Just going to drive them through here, which is actually going to cause the turn. It's like a rotor, and it's going to phosphorylate your ADP, okay, into ATP. So it's going to use the energy from those protons to add a phosphate back on ADP and turn it into ATP, creating the energy. So it is also, we call this chemiosmosis, chemiosmosis, okay, that proton motive force that is driving this process, okay, to turn and create that ATP. And then we're going to have our oxygen as our final electron acceptor here, and we have some hydrogens here. They're going to be bond together and pick it up and turn it into water. Okay, and so that's why breathing is so important, because if we don't pick up the hydrogens down here and bond them to something else, what's going to happen? They're going to build up on this side of the membrane. If they build up on this side of the membrane, what's going to happen? Well, I've got a high to low concentration. Well, if this builds up over here, I don't have a low concentration over here anymore. We'll reach equilibrium, and what will happen to this whole process? It'll stop, and we will no longer be able to do aerobic respiration. So that's why that final electron acceptor is so important. All right, so those protons or hydrogen ions are now in the cytoplasm. They've come across the membrane for that final process that we were just talking about. This is the picture in the textbook, so looks a little different. So I wanted to kind of relate this to the picture that you have in your book. So this is the final step now. We've taken our electrons, and they have gone down this chain here, these enzymes here in this membrane, and we are pumping hydrogens across. Electrons are flowing down, okay? And so the hydrogens are coming back through that proton motive force or chemiosmosis, and oxygen, and this is where this one is showing it. You can see oxygen can pick it up, but here are our other electron acceptors. Final electron acceptor or terminal electron acceptor means the same thing, okay? So they're moved via that chemiosmosis, and that's providing that energy for this ADP, adenosine diphosphate, to be phosphorylated, which means adding the phosphate group. So there'll be a little inorganic phosphate group floating around here, okay? And so then that's going to be converted to ATP, which makes all of the energy, a total of 36 to 38, okay, per whole session. You'll make about 12 waters. All right, sometimes it'll say 6. It kind of depends on how they're counting it, okay? And so then you have your nitrates and sulfates that can pick it up as well. So aerobic respiration right here, and then anaerobic respiration right here, okay? In eukaryotes, this is going to occur on the cristae. The cristae, remember, are these is this inner membrane of folding. And so here you can see this is the inner membrane or extra membrane space here where the protons go, and then they got to flow back across that membrane. And so the coupling or putting together of linking together the ATP synthesis, we call that coupling, and the electron transport chain is what we call oxidative phosphorylation, because we're using oxygen to phosphorylate, and we're also using oxidation, all right, oxidation to load NAD and FAD with those electron carriers. We're using the oxidation reduction process here, okay? And so that occurs on the cristae of the mitochondria for eukaryotes. So there's a side reaction that can occur in the electron transport chain, and that is the incomplete reduction of oxygen to those I want to say caustic, to those reactive forms of oxygen, toxic forms of oxygens, okay? So superoxide, and that's the O2 negative, and hydrogen peroxide, which is the H2O2. Remember, those are toxic, and oxygen products that are harmful to cells, okay? One exception to this, by the way, is Streptococcus. It grows well in oxygen, but it is not able to produce catalase to deal with these particular substances. But remember, we have, and many other organisms have, superoxide dismutase and catalase to help deal with these toxin-oxygen products. Okay, so remember, we can break H2O2 down into water and oxygen. Okay, but Streptococcus species of bacteria are not able to do this. Okay, let's see what else. All right, so anaerobic organisms are not able to handle these oxygen products, which limits their ability. This is what limits the ability to survive in an aerobic or oxygen-filled environment, because this will eventually build up and will cause them to basically die, or the cell could rupture. It just kind of depends on the mechanism. Okay, so in anaerobic respiration, the final step, or the terminal step, is going to use inorganic oxygen-containing ions instead of that free oxygen as the final electron acceptor, and we kind of just looked at that. But nitrate reductase is an enzyme that can actually help remove those toxic forms of oxygen, or to pull oxygen from nitrate. All right, so it catalyzes the removal of oxygen from nitrate, which turns it into nitrite, which just means it has one less oxygen and water as products. Okay. We also have denitrification, which sounds complicated, but it just means taking off a nitrogen. That's an important step in recycling nitrogen for the biosphere. So these guys that can do this are really important ecologically. Some species like Pseudomonas and Bacillus have enzymes that can further reduce nitrate to nitric oxide. So they can pull another oxygen off and turn it into nitric oxide, which is NO, or nitrous oxide, okay, or even nitrogen gas into. Okay, really important to recycle, for that recycling of nitrogen in the biosphere. You remember from elementary school, had you learned about the water cycle? Well, we have nitrogen cycles in that as well. Okay. Other oxygen-containing nutrients are going to be reduced anaerobically by other methods, or by other bacteria. Those can be carbonates or sulfates, okay, and that's that CO3 2 negative or SO4 2 negative. None of the anaerobic pathways produce near as much ATP as aerobic respiration. It is just much, much more efficient. So that third step was fermentation. And remember, fermentation is the incomplete, or the third pathway, I should say, incomplete oxidation of glucose or other carbohydrates without oxygen, okay? So we're going to have these organic compounds as the final or terminal electron acceptor. We're going to make a very small amount of ATP. Fermentation is not near as efficient as aerobic respiration. This is why you can only do it for so long in our muscles. We have to, at some point, our muscles are going to give out because we're going to run out, okay? And so organisms that don't have an electron transport chain will definitely utilize fermentation. But we have some very adaptive guys that can actually repress the production of those proteins in the electron transport chain if oxygen is lacking in their environment. So they can kind of flip the switch over and revert to fermentation, which isn't near as efficient, but allows them to survive for periods of time without oxygen, which is a pretty handy trick. And so we have two types. We're going to have our alcoholic fermentation, okay? This is where you're going to make ethyl alcohol. You're going to have ethanol and CO2 as the products. We're going to have some acetone and butanol as some solvents here. We're going to make some organic acids for this portion over here if we do lactic acid fermentation, okay? So we can also produce lactic acid, which is where we get that lactic acid burn, at least that's partially correct. And then we can also make, or we don't, but organisms can make things like acetic acid. And acetic acid is actually like vinegar. That's what vinegar is. It's acetic acid, which is pretty neat, okay? Let's see. And so the organic molecules that are reduced in this process, we actually can take advantage of those. They're often very useful. It's going to be things like the ethyl alcohol, which this is how we make wine and things, lactic acid, propionic acid, butanol, other things like that that we really take advantage of, all right, as human beings. Um, I think that's it on this slide. So some of our other compounds that aren't carbohydrates go through catabolic processes as well. Things like complex polysaccharides. Those can actually be broken down and then changed into sugars that can go through glycolysis. Lipids, of course, we know we get energy from lipids, and so they can go through glycolysis as well. They're broken down by lipases. Remember, ASE is an enzyme. And then those can go through a different area in the process, and so they kind of enter at a different point. Fatty acids can also they enter in Krebs cycle. So all these other things that we eat that we get energy from, they still go through aerobic respiration for us, and these other anaerobic processes, they just enter at a different point instead of at the very beginning. And so glucose we get is what we use the most because it goes through the whole process, but other things work as well. Okay, they just enter in a different place. Okay, proteins are going to be broken down into amino acids. They're going to go through deamination, which just means removing an amine group, and then they're going to be recycled and put into the Krebs cycle because you can break them down into intermediate portions that fit into that Krebs cycle and enter there. So this is a good overall look at those 4 different things and where they kind of enter. You can kind of see here what I'm talking about when they say they enter at another portion in the cycle. Cells have mechanisms for careful management of their carbon compounds. Catabolic pathways here, like we said, they have several intermediate products. So if I'm breaking down, for instance, this protein into an amino acid, I can deaminate that. I can skip glycolysis here and just go straight to this portion right here, which is that intermediate step, okay, and go on into the Krebs cycle. All right. Sometimes I can even skip down here and go all the way to this portion of the Krebs cycle. All right. You can see here fatty acids can do the same. They can skip and go all the way down here. So they just enter at a different location. All right. So these are different metabolites that can be kind of diverted into other pathways. They can also go to anabolic pathways. And so a given molecule can actually serve several purposes. And so maximum benefit can be derived from all of the nutrients that we eat and all of the metabolites that are going to be used by us or by these pathogens and other microorganisms, okay? So we also have what we call amphibolism. Amphi, remember, is kind of like ambi. So that's the ability, like ambidextrous, the ability of a system to actually integrate catabolic and anabolic processes to be the most efficient that the cells can be. All right. So being able to use several sources there. All right. We could have 2 sources, say, for monosaccharides, amino acids, fatty acids, nitrogen bases, and vitamins. They can enter the cell from the outside as a ready-to-use nutrient, or they can be made through various other cellular pathways. And so the degree to which that organism can synthesize or make its own building blocks that it needs for, say, nucleotides or amino acids to make proteins is kind of genetically determined and varies from group to group, okay? But always remember, glucose has that central role in metabolism and energy, using energy, and the main component of cellulose in the cell walls and some eukaryotes have storage areas where we can put things, okay? We also have those intermediate products that we can use. Peptidoglycan is a really important thing that we can build from fructose 6 phosphate. Glycogen is built from glucose 6 phosphate. And so remember, we can use these to drive metabolism, but we can also use them to build things up as well. That's the key understanding of this slide, okay? We can use these processes to gain energy, or we can use these as building blocks to make something else, okay, to build. Anabolic is, and so that's what's up here at the top. This is the anabolic reactions where we can take these products, we can take and make a nucleotide, which is the monomer for a nucleic acid, to go on and make chromosomes. We can take amino acids and make proteins. We can take carbohydrates and build cell walls out of them, or make starch or cellulose out of them. We can take fatty acids and help to build that phospholipid membrane. We can store them, or we can go and we can use them in a catabolic reaction, break those down and make energy with them. So kind of winding up those anabolic processes, we're using those to assemble the cell. So components of bacteria are going to be synthesized or made continually. And so we have catabolic reactions and anabolic reactions occurring simultaneously all the time. Catabolism is also taking place as long as those nutrients are present and the cell is not dormant, okay? And so then we have these anabolic processes where we're going to make those macromolecules. We've got DNA replication going on. Membrane and cell walls, we have growth and reproduction going on, okay? Catabolic processes provide all the energy to do these complex building reactions.